Before jumping into this this discussion, whenever we actually get different people from different backgrounds together, it's always good to try to find common grounds. And this is my starting point 2,000 years ago. So we'll do 2,000 years of history in just a slide. But when we're talking about public health, clearly we're thinking that epidemiology goes hand in hand, and we're thinking of advances in biomedical sciences to be essentially the action arm of public health. But what I would like to submit, being an environmental engineer as well as a microbiologist, that another important action arm of public health is environmental engineering. And really this story started 2,000 years ago with the Romans, who built very large sewer systems, cloaca maxima, built latrines, built aqueducts. They actually knew that water was at the center of the health of their population. And they built infrastructures to do that. And then we kind of forgot about it until major epidemics in the 19th century. And what I want to say is John Snow here, who's seen as the father of epidemiology, interestingly, is also seen as a father of modern sanitation and one of the heroes of environmental engineering. So John Snow single-handedly was able to identify that it was a water distribution pump in the middle of London, the Soho district, that was responsible for spreading the disease. And this is how one of the pieces of the puzzle that led to major build of the London sewer and eventually wastewater treatment. And then if we come to the 20th century, essentially the increase in life expectancy, of course, biomedical science and advancement over the last century would claim a lot of credit, but environmental engineers would also claim a lot of credit in that achievement. We would say, among other things, that drinking water treatment and wastewater treatment contributed quite a bit to the prevention of diseases, as we can see here in Philadelphia. The cases of typhoids decreases immediately upon the installation of drinking water treatment. So essentially, environmental engineering and public health go hand in hand. So I want to tell you a little bit about environmental engineering, what we do, and in the case of wastewater, essentially we build infrastructure to ensure chemical and biological safety of discharged wastewater. So as we go along, we produce wastewater, and the goal of wastewater engineers is to treat it to make it safe chemically and biologically. The new vocation, though, in this modern sustainable development era, is to say, well, we don't only want to treat the water, but we want to recover resources. So as you will in the future, the industry leaders will talk about not wastewater treatment systems or facilities, but rather water resource recovery facilities. And we recover water, fertilizing nutrients, energy, and carbon. But in this context of epidemiology and understanding the disease, what else can we recover? And we realized, in fact, we have a lot of information. There's a lot of information that we actually flush down the toilet. And this is recovering this information that is the basis for wastewater-based epidemiology. Now, this is not a new idea. There is actually good success stories in the past. So polio, for example, has been surveilled using wastewater for the last 25 years or so. And for example, there's a case in Israel where in 2013, polio was actually detected in wastewater, which actually led to immediate action by the authorities and then a rapid curtail of this outbreak. We're also moving along. Different people are looking at chemicals, and recently Statistics Canada, with the help of Viviane Arjot at McGill University, put together a pilot project to look at the consumption of illicit drugs and measure consumption in different cities around the country. And we see here the results that they published last year. So they're following 14 wastewater treatment plants, about 8 million Canadians. And we can see that, well, interestingly, if we compare cannabis and methamphetamine, well, different Canadians will have different consumption profiles, or different cities will have different consumption profiles. So again, information, and we're moving along to obtain that information in different ways. So developments that are going on right now, secreted drugs, the same thing that we do with illicit drugs, we could do with licit drugs, or antimicrobial use. Most of these are very well regulated in Canada. It's not true around the world. So tracking from the sewer the use could be interesting. Clearly, when we're talking about wastewater, we're trying to prevent enteric illnesses. So enteric viruses and bacteria are more and more tracked, and there's techniques that are being developed. And there's also novel applications that are being developed right now. So my work, one part of my work is to develop technologies to track antimicrobial resistance in sewer systems, especially for resistance in healthy populations, so populations that are out of the clinic. And there's actually a number of viruses that are not necessarily known for a major fecal oral transmission, but they actually are present in our feces and in the wastewater. So influenza is one of them, coronaviruses, and among the coronaviruses, clearly SARS-CoV-2, the agent of COVID-19, is the one that interests us today. So when we're trying to look at the virus in wastewater, the virus could be in different shape, and different questions could be asked, and being interested to get more information on. So the virus could actually be discharged intact and stay alive in the wastewater. If that is the case, we can ask, is there a fecal oral or fecal respiratory transmission? To the best of my knowledge, there are no reports so far of a fecal oral or fecal respiratory transmission. We know it's present in the feces. From time to time, scientists have been able to isolate live viruses from feces. No live viruses have been so far isolated from wastewater itself. And the only case of fecal respiratory transmission with SARS is during the 2003 epidemic, where a Hong Kong building, essentially that's a very specific case, the sewer pipe in the building was leaky, and the sewer was the waste was actually leaking in the ventilation system of the building. So which is very, very different than what you would find, of course, at the end of the pipe at a wastewater treatment plant. So in that sense, the best information so far is it's safe for wastewater workers to work around wastewater today. Well, there are already a lot of viruses and bacteria, harmful bacteria pathogens that would actually likely affect them. And we don't think that SARS-CoV-2 at this point would be an extra danger, but it would be nice to get a bit more data on this question. So the virus could be intact and alive, but it could also be incapacitated or partly degraded. Even if it's partly degraded, we can potentially still detect its RNA, and we could potentially also detect its proteins. So both have been detected. I will mainly talk about the detection of RNA for the rest of the talk with similar techniques as we use for clinical diagnostics, the RT-PCR, reverse transcriptase quantitative PCR. But one of my colleagues at University of Ottawa is in a group, and they're detecting the protein themselves in wastewater. And eventually we're gonna have to see if it can be quantified, because the basis of a lot of what I'm gonna be talking about today rely about this capacity of quantifying accurately the RNA in the wastewater, or the virus in the wastewater. Now, the viral material, including the nucleic acid, including the RNA, could actually decay. And it will decay faster at higher temperature. So my little graph here shows that at high temperature, you lose the signal very quickly. At lower temperature, a little bit less. And that is very important, because our wastewater will spend some time in the sewer network. And that will actually affect both the signal that we recover and its interpretability. We will come back to these questions as we go along, but these are very important challenges for the interpretation of the results that we obtain. So how do we detect the virus? Essentially, we use a very similar diagnostic technique that is used for clinical samples, this reverse transcriptase quantitative PCR, where we extract the RNA and do the transform to the DNA and do quantitative PCR on it. Now, there's a slight difference, and it's often this difference is often present between application in clinical settings and environmental settings. Because often when we do diagnostics in clinical settings, we really care about a presence-absence test. So is this individual, can I detect SARS-CoV-2? Therefore, he's affected by, the individual is affected by COVID-19 or not. Well, in order to really provide information that is useful in environmental samples, and specifically here in wastewater, we actually need to obtain good quantification. We don't need to know if the virus is present or not. We need to know really how much of the virus is present and how does it change over time. So that is one of the major differences. What are the potential advantages of detecting the virus in wastewater? Well, potentially we could actually detect the virus earlier than when we actually wait for symptoms to set on and test after that. We could also detect asymptomatic or pre-symptomatic cases. And we have potentially the capacity to track the entire population at once. So what is the information that we can recover and provide to public health? And here I've put it in 3 cases, and I have to thank the international community for these. This is coming from the Water Research Foundation summit that we had a few weeks ago. And essentially, we could actually track the concentration, the titer or the load of the virus in wastewater. So we can detect maybe an increase, and that is probably relatively easy to achieve. We could potentially detect the increase and eventually the decrease to see if our measures work. Although there are some caveats and challenges associated with these two trends together. And that's probably the best that we can do today, or relatively rapidly. Now, early on, you may have heard that people try to evaluate the community prevalence. There's a study that came out of Boston that was trying to do that. Really, if we think about it, it is very difficult to transform this viral load into the number of cases in the field. There's a lot in making that calculation, and hopefully you'll have a bit of a flavor for this as we go along. So, has it been demonstrated? Can we actually detect SARS-CoV-2 in wastewater, and is it related at all to cases, clinical cases? Well, the first demonstration came from this study from the Netherlands, where they studied in the month of March 6 municipalities, and we actually see that we see a relatively good correlation between the number of cases and the number of gene copies in the wastewater. And this is from this study where you've heard the number of a detection limit of about 1 per 100,000 cases, or 1 case per 100,000 of population. And so I'm going to give just 3 examples, but this other study from France, they essentially looked at the number of cases in Paris wastewater treatment plants, number of viral genomes in at wastewater treatment plants around Paris, and they looked at the number of new cases and cumulative cases. And essentially, this dotted line on the first panel is an estimate of how much virus was probably shed at any moment in this wastewater. And we again see that the dynamics is relatively following the same trends. Now, if we want it to be an early detection limit, could we actually show that it's the titer, that the concentration of virus increase faster than what we see in reported clinical cases? And New Haven, Connecticut, this last study here, essentially in red you see the viral RNA, and they tracked both the hospital admissions, but also the COVID-19 cases. And if you do, so in the first panel here, the red line, so you have a lot of data, and they do a moving average, and this is this red moving average that is reported in the second panel. And if they do the same thing with the number of COVID-19 cases, essentially the peak is obtained in the wastewater about a week before the COVID-19 new cases. So it does seem that it could act as an early warning system. However, I was talking about quantification, and let's look at the data reported in all these studies. What we do see, notice that these first 2 graphs here are in log scale. We do see a very large scatter. So the data tend to be very noisy. Now, these were measurements that were done at the end of the wastewater network. We could also do measurements within the network. This is the island of Montreal. So we see around the Ottawa and St. Lawrence rivers. And if I just change the map for you, we can easily see the different collectors essentially all the wastewater goes into these collectors. We could actually measure at the end of the network at the treatment plant. But we could also, if I go back, all these points are points where small neighborhoods collectors are actually getting to these main collectors. So these are actually major points where we could take samples and follow various neighborhoods. So we can actually vary our granularity. Now, these are the possibilities. What are the challenges? What are the difficulties? Well, I just want to, if you notice here on the map of the island of Montreal, we have this region in green and this region in beige. And it's because essentially we have two different sewer systems. In the regions that are green, essentially we separate the rain and the sewer or sanitary water. So that makes a sanitary water that is a lot more concentrated. Whereas in the region that is beige, this is the legacy of older treatment systems or older sewer systems. The rainwater as well as the sanitary wastewater is mixed. So interpreting at the end exactly what does the signal mean and compare it eventually between municipalities, you can see right there, could be very difficult. So here are some other challenges. So in order to be useful for public health, we need to be able to make a judgment relatively fast. We need to judge a trend in this noisy data in a few days. And that's not totally clear that that has been really demonstrated yet. And that is very different. That's a new challenge for wastewater-based epidemiology. If we talk about the polio tracking or surveillance and the illicit drug example, well, these surveillance and studies actually have the luxury of time. You don't necessarily have to make a decision within a few days. Here, we actually need to make a recommendation to public health to go intervene, to go look what's happening in this or that neighborhood within a few days, not within a few weeks. SARS-CoV-2 is an envelope virus, meaning that it's hydrophobic. So it's been difficult, and lab people like me are in discussion with this. It's been difficult to have protocols that consistently recover the genetic material correctly. And as the virus goes through the sewer system, the virus will decay, and the flow, the weather, the temperature, the amount of solids will actually affect the decay of the genetic material. This is actually a modeling study, so these are not real data, but that gives you the drift of it. So the temperature changes over the entire year. So we have here from March to July. And what we see here is if I want to detect a viral load of 100 units are not important here, 100, how much would I need that it's emitted by my population? And the increase here is the increased amount of travel time. In the map that I showed you of the island of Montreal, essentially the entire network, the bigger collector, is about 40 to 50 kilometers away from the, actually more than that, about 100 kilometers away from the treatment plant. So the wastewater will take a few hours to travel. So in Detroit, where the temperature does not change too much, the difference between winter and summer is not that much, but still you see a seasonality, which eventually we'll need to take into account if we want to interpret. And in a place where it's very warm, like in Tempe, Arizona, we see that the impact of traveling time and of season is much more important. This is actually, if you consider, so this is Tempe, Arizona, one of the sewer network. If we consider the travel time of different places in the network, they all drain to this cross here. Essentially, we see that the furthest from this draining point, there's only, in the summer, there's only 80% of the emitted charge that is remaining when we sample here, whereas much closer, we have much higher residual signal. We saw different sewers will have different infrastructure, maybe different municipalities will have different industrial contributions. And on top of that, this is only on the environmental engineering side. We also know that for some patients, they tend to actually shed the virus for a long time after they're not positive anymore for their swab. So don't worry about the early part here. There's no data, so we cannot know if the feces samples were positive before the throat swabs or not. But what is important is that we actually see for some patients that they actually still shed the virus several weeks after they're recovered. Nonetheless, this line of research attracted a lot of work internationally and in Canada. So far, there's certainly more than 20 papers from a lot of different places. We're aware of a lot of unpublished studies, and there's in Canada a number of groups from across the country that are working. There's also a lot of international collaborations, but also major groups nationally driving important efforts. So in the Netherlands, there are 37 cities in a demonstration study. Germany also has a number of investments, strategic investments. So I'm just here mentioning a few, but it's just to show that there's a number of places where at the national level, there was strategic investment in trying to develop this effort for public health. What I want you to notice also at the bottom here is that the wastewater or the water industry has major players, major groups bringing the industry together, and they are also very interested in driving these efforts. Similar things happen in Canada. So the Canadian Water Network, about a month ago, created this coalition for wastewater-related research on COVID-19 with the hope of educate and link better public health officials, wastewater utilities, and scientists, and to try to organize the entire landscape to have a pilot study and demonstrate maybe the use of this approach. Now, we've been at work in this coalition for a while. We developed a number of principles to guide the design of the pilot studies. But one thing that we realized is that at the point 8 here, we should consider, if we go to smaller resolution, so neighborhoods and maybe institutions to monitor, well, that will actually raise a number of ethical problems. So these are things that we also need to address. How much does it cost? Well, this is a few numbers here that I've assembled from different sources. But if we wanted to test the entire population, essentially just looking at Canada and our current, my estimate of the current testing capacity, It would take about 22 months to test all Canadians and would cost nearly $1 billion. And let alone the problem of false positives that would creep into this analysis. Well, if we wanted to do everyday an analysis of wastewater and test or track about 80% of the Canadian population, we would run about 2,000 to 3,000 tests, and on a daily basis, it would cost about $60,000 on a daily basis. So much cheaper, and we could actually track most of the population. There are ways to also reach out to Native people and also track them better with that kind of techniques. So in finishing, I'd like to thank people in my research group at McGill and the number of collaborators that about a month ago, about 2 months ago, we started to work together on a lot of these issues. The Canadian Water Network and the Scientific Advisory Committee of the Coalition. And I'm also a member of Centro, which is a specialized Quebec water research center. And they're very helpful in all these endeavors as well. Thank you.
That's wonderful. Thank you so much for sharing. It looks like we're at our half-hour time, so we won't have time for questions today, but you have been thanked for a very informative presentation, and thank you for sharing. If anybody wants to reach out to Dominique Frigon, you can reach him on Slack. And you can also join the Theme Viral Detection Sewer channel on the Slack platform for CanCOVID. Thank you everyone for coming. Thank you so much.
Thank you.